Hello, I'm Sanskrita, and I'm very interested in understanding what makes or breaks our immune systems. Cancer is one such question, and hence my topic. Fun fact, in the background of this slide is an image of PD-1 expression on T cell surface as seen by fluorescence microscope. We will discuss a little bit about the PD-1 receptors in the upcoming slides. So, moving on. In the duration of the next few minutes, I will be speaking about immunotherapy, specifically immune checkpoint inhibitors in the context of non-small cell lung cancer, and also discuss the results of analysis of gene expression during therapy. So first, about non-small cell lung cancer or NSCLC. When we look at the diagnostic and mortality figures of cancer, lung cancer trumps all other types, causing almost 20% of the deaths, and non-small cell lung cancer accounts for 90% of these deaths. Therefore, a rigorous study of NSCLC and our therapeutic options are very important. Currently, the doctors prefer surgical resection and radiotherapy for early stage patients, while chemotherapy is provided to late stage patients. Our last confirmed data pegs NSCLC deaths to a close 1.62 million. Clearly, we need improved therapeutic modalities. So, immunotherapy. Immunotherapy manipulates the immune system's own effectors to deal with these disease conditions. I'm going to focus more on the immune checkpoint inhibitors here. So the receptors program death 1 or PD-1 and cytotoxic T cell associated protein or CTLA-4 are expressed by almost all types of T cells dynamically across time and magnitude of immune response. So in normal conditions, they would interact with their ligands on dendritic cells and suppress the immune response. This forms the basis of autoimmunity prevention, which is essential. But cancer cells, taking advantage of this, also express ligands PD-L1 and B7, which can help them to mute the immune response when it is actually required. The interaction between PD-1 and its ligand sets off a cascade. It causes effector T cell dysfunction, exhaustion, and neutralization of mature T cells, prevents further activation of naive T cells, and also can activate the regulatory T cells. All these together produce a cytokine brew that makes the tumor microenvironment highly inflamed. The tumor growth becomes more aggressive and invasive, gradually shutting down all branches of immune system. So immune checkpoint inhibitors interfere in the first step of this process, blocking any interaction between the receptors and their ligands by competitive binding to one or the other. Even in the league of tumors, NSCLC shows extremely high mutational burden, so it becomes very affable to this form of intervention. The data that I have analyzed is trial data of checkpoint inhibitor nivolumab. It is a humanized antibody that binds specifically to the receptor PD-1. Although it is theoretically a very robust therapy, many failures have been observed in the form of adverse immune events and or progressive disease. So the two questions I had going into the study was whether genes expressed in cytotoxic T cells have any influence over the response towards nivolumab therapy, and if administration of nivolumab influences the gene expression in these cells. So I got the data set from Geo, and here is a summary of the available information. So most of the patients had already tried either surgery, radiation, or chemo and did not see any improvement. So they were put on a course of nivolumab, and their serum samples containing peripheral blood mononuclear cells or lymphocytes extracted before administration and 6 to 8 weeks after the administration. So before we move further, a small point needs clarity. The response here refers only to partial response throughout the study. In case of lung cancer, we generally can't hope for anything better than that. Now we can talk about the steps taken to answer the questions I had. So the sequence reads were found to be multiplexed, and they had to be demultiplexed before alignment and mapping. Bouty 2T and RSM expression table algorithms on the T-BioInfo platform were chosen for alignment, mapping, and generating read counts. So once the expression data was obtained in expected counts, genes that had low average counts were eliminated before performing further analysis. Then, based on the questions I wanted to answer, I created different groups of data for exploratory analysis and differential expression analysis. So, on to the results of these analyses, starting with PCA for exploratory analysis. The first graph seen here is for a general analysis of how the data segregates based on expression values. This file contained information regarding responders and non-responders labeled at time points before and after the administration of nivolumab. So the only slightly clear clustering is seen in the violet dots on this side of the graph, with one outlier here. And these are from non-responders at the final time point. This does not seem to be very useful for us, and an outlier excluded PC analysis could be performed, but this data is spared. So excluding this outlier would mean that we would have to exclude some green point as well. This is a small patient group, and this does not seem to be a very optimal solution. So I went ahead and tried to analyze the expression values as a whole at the initial time points and final time points, and between responders and non-responders with no clear distinction again, to see if nivolumab influences expression in killer T cells. A large PC analysis was performed for just the responders between initial and final time point. Even this does not provide any insights into the relationship between expression values and the known phenotypes. However, if we look at the first three PCA plots, this one and the first two in this page, we see that there is a very distinct separation between the data points right down the middle for reference. So this clustering is not explained by any of the groups or definitions provided by the data. This points to some missing sample information that could help us understand the interaction between nivolumab and CD8 positive T cells better, but it's not available at the moment. Now, on to the differential gene expression analysis. So the first analysis was performed between responders and non-responders at time point N1 to understand if gene expression could somehow influence the therapeutic response. Here is a volcano plot to represent the downregulation and upregulation in genes with their significance. So to the left of the graph are the downregulated genes, and to the right are the upregulated genes. Lower negative log P values indicate low significance. And as we can see, most of the genes have a negative log P adjusted value of below 1. And the actual P adjusted value is very high. And upon a preliminary sorting of the output table, the smallest P adjusted value came out to be 0.13, which is much higher than the preferred 0.5 threshold. So this finalizes the fact that differential expression in responders and non-responders before administration of nivolumab is not significant enough and hence cannot possibly influence the therapeutic response. Then the second group of analysis were the expression in killer T cells of responders between the initial and final time points. We are looking to see if nivolumab influences the expression in its course of action. So one glance at the volcano plot shows us that it does, not by a lot, but it does. Only 3 significantly downregulated genes were observed, and these results prevent us from moving ahead to analyze pathways and gene ontology terms that are differentially expressed. But these 3 genes are also involved heavily with many important cancer cell death and immune response pathways. All of these genes produce long non-coding RNA and are extremely essential in the regulation of protein coding pathways, although they are unlikely to be translated themselves. So block 1 is transcript is shown to be involved in 2 important pathways, and its downregulation is associated with silenced T cell factor 4. T cell factor 4 proteins, along with other proteins of the TCF-LEF family, are directly involved in inducing the transcription of genes downstream of the WNT pathway. So the expression of these genes influences T cell development and differentiation at various time points and tissue types like thymus, lymph nodes, and tumor microenvironment. These gene products can act in both stimulatory and inhibitory ways. And a decrease in stimulation of WNT signaling pathway improves naive CD8 positive T cell conversion into mature cells and reduces FOXP3 positive transcriptional activity in regulatory T cells. Hence, the downregulation of block 1S, as seen by the graph, can deal with effector T cell exhaustion and improve T cell activation and reduce immune suppression in the tumor microenvironment. So, TC4 silencing also directly improves anti-tumor activity of antigen-presenting cells and thus can help in delaying cancer growth. Block 1S is also associated with ferroptosis, which is different from apoptosis and necrosis. It is a novel cell death pathway, and we won't be going into the details regarding that. However, proposals have been made to use genes associated with ferroptosis as diagnostic and prognostic markers in breast cancer and a few other tumor types. So, studies for its use in lung cancer prognosis have not been conducted yet, but this could be a good starting point. And then coming to the next gene, the TMED7 TCAM2 could be the whole locus near this gene is associated with the TRAM-TRIF pathway. A downregulation in the TMED7 TCAM2 could be associated with reduced inflammation in tumor microenvironment, and this could reduce the number of regulatory T cells near tumors and further prevent the conversion of helper T cells into regulatory T cells. For a bit of clarification, although the study analyzes expression in cytotoxic T cells of adaptive system, the responses of adaptive and the innate system are usually influenced by cells from either of the systems in our body. So, yeah. Anyway, about the third gene, not a lot of information is available about it for now. Hopefully, we'll be able to study how it factors into all of this soon. Now, here's a recap of the questions before we summarize the results. The PCA plots show a distinct separation undefined by available data. There seems to be some confounding variable influencing the separation based on gene expression. And based on differential analysis, gene expression in tumor infiltrating cytotoxic T cells cannot influence response to nivolumab and cannot be used as a marker for predicting the response either. However, nivolumab does affect gene expression in these cells and can be used as a marker for analysis of disease progression. I believe that integrating other omics big data could give us better results and find important prognostic markers, but the expression library used also seems to be a bit lacking. As we saw in the mechanism of PD-1 action, inclusion of other cell types like helper T cells and regulatory T cells could possibly fill this gap. And after some discussions, the time duration between measurement of response could be one of the factors causing less differential gene expression. Like 6 to 8 weeks between response measurement, according to RECIST guidelines, apply for tumor cells. The change in gene expression of T cells precedes the tumor reduction that it would mediate. So a few weeks before this time or increasing the frequency of sample extraction could improve the study greatly. And using different algorithms and analytical tools is always an option. But the probability of that alone improving our results is very slim. So in conclusion, dealing with lung cancer is still not in a stage where we can confidently look for cures. The clinicians are currently focused on controlling growth of tumor and palliative care. And immune checkpoint inhibitors show promise on paper and also in many trials. But to make up for the dearth of response, use of combination therapies is growing more popular. And the second regimen in the combination therapy being of chemotherapeutic agent like cisplatin or other immune checkpoint blockers like atezolizumab or ipilimumab. So till these trials give us adequate results, tobacco eradication must be our number one step against lung cancer. That is it for my report. And I'd like to take some time to thank my mentors and guides, Dr. Raghav and Sonalika ma'am, for helping me put this together and working through this project. Thank you so much.